Good morning, everybody. My name is Anand Shrivastava. I'm a lawyer at Williams & Shire Environmental Lawyers. Hope everybody's well this morning. Thanks for joining us as we talk about sulfolane contamination, and we have a handful of very interesting topics to cover in the next couple of hours. Just at the outset, I'll ask our friends at ESA to introduce Tara Strike, and I understand they have a small message for everybody.
Tara Strike is based in New Hampshire. Our manufacturing facility is outside of Toronto in Brampton, Ontario. And we have done work in 5 nations throughout the world. The majority of our work is in North America, Canada, and the US. Been in 23 states and 4 provinces. We've also been in Japan. We've been in the tannery districts of India. And we have done sludge work in Saudi Arabia.
Well, this is messed up. So this isn't his name, right? And everyone's calling him a woman, right? So at this point, he's like,
Thank you very much to Tara Strike for generously sponsoring this session, and let's jump right into it. We have 4 very interesting topics relating to sulfolane, and in the interest of time, the way that we've set this up is that there will be about a 20-25 minute presentation, and from there we'll leave about 5 minutes for questions, and we'll do that 4 times over. I encourage you to please submit your questions into the question box via the GoToWebinar platform. I can see those on my screen, and I'll pop back from time to time and ask the questions at the end of each presentation. We're going to start with correlations between routine parameters and sulfolane, as you see on the screen, and we really appreciate Deanna and Paul offering their time. So I'm going to stop there and turn it over to them.
Thanks a lot. Everyone can see my screen, I hope. Had a few technical difficulties this morning, so hopefully we've made that work. Small introduction. So myself and Paul both work for Shell, and we have been working on sulfolane for the last couple of years, and so we wanted to share a lot of the work that we've been trying to progress over the last couple of years. While we're using it internally, we wanted to make sure that anyone else who was working on sulfolane had the opportunity to see it. So please do ask questions. There are 4 different presentations, 4 different aspects of things we've been looking at that are all linked. If you have questions, I encourage you to ask them whenever you like. And if it happens that you think of a question about a previous presentation, please just ask it in the next one. This is not really intended to be 2 hours of Deanna and Paul talking with no breaks. So with that, we'll get started, hopefully. All right, so this is a bit of a recap. What do we know about sulfolane and some of the challenges associated with the properties associated with sulfolane? It's not volatile. You can't smell it. You can't use a gas tech to find it. You can't use PID. That makes it challenging to know if it's present. It's very stable, doesn't really react well with a lot of other things, and doesn't degrade anaerobically very well. It has a high selectivity towards aromatics, so it is happy to go wherever a hydrocarbon plume may be. It also is happy to go in its own direction. And so while it's both polar and non-polar, that makes it a challenge to predict where it ends up. It's very soluble in water. It's not like when you've got an apple in a well, there's no phase separation that you can see. It's colorless, so it looks like water. So all these tools that we're used to using in water and soil that gives us some visual or olfactory indication that there's sulfolane or a chemical there, we didn't have any for sulfolane, which is really quite frustrating because if you don't know you have it until you've taken the sample back to the lab, it costs a lot more money and it's a lot harder to get it first time right. If you compound the fact that there's no indicator available right now to identify sulfolane with the fact that it behaves differently depending upon what water matrix it's in, it also can behave differently because of density gradients depending upon how fresh the water is. Then at your site, your water matrix can change over time, which doesn't help because the behavior of sulfolane also changes with your water geochemical changes. By the time you collect a sample, get it back to the lab or research facility or something, the physiochemical properties of the water have probably changed because it's been exposed to oxygen, or in order to do any experiments, you have to expose it to oxygen, which also makes it challenging. And then how do you try to design an experiment that's going to take into consideration all of the challenges that we just talked about and still figure out if your indicator compound, even if you could find one, will work and work reliably and repeatably, regardless of whether you're in surface water, groundwater, brackish water, fresh water. That's what we were trying to do. Ambitious, potentially. So how did we go about it? One of the first things we did was we created a bunch of different synthetic waters. So knowing that going to our sites and grabbing water, the shelf life of that water was quite short. And so what we tried to do was create stock solutions, if you like, of groundwater and surface water compositions that were representative of the waters that we encounter at some of our sites. And this sounds really simple. I thought it was really simple. Just add in the chemicals that are in the water. It sounded very simple. If you do that, you'll get a really big sludge really quick, and it doesn't do you any good. So what people do typically in the labs is they have some stock solutions of representative water, but they don't actually try to create water that is synthetic groundwater. So we did that. That took quite a while. It took lots of monitoring, and then it took months of making these stock solutions and then adding them together. So instead of simply adding sodium and magnesium to get the right concentrations, you have to make solutions that will not have ancillary, secondary, or tertiary reactions with each other when you add them together. Took a lot longer than we thought, but that's where we got. So now we have these synthetic waters that we feel are fairly stable and can be used in the lab and represent several different scenarios of actual groundwater. So that's the first hurdle. So now all we've got is the base groundwater. We haven't got to the indicator part yet. This is the water quality variability that we had. So we tried to take kind of a surface water example, some sort of till that's very Albertan, and then a fractured bedrock. And we took well over 150 samples. We ended up taking geometric means for all of the different samples that we took and created these representative groundwaters. As you can see, TDS varied quite a bit, sulfate varied, conductivity varied. That was the point. We were trying to create things that would look like real world. So now that we found water, what do? We've been told anecdotally by a lot of our old time operators, if you're looking for sulfolane in the water, just look for a pH change because you know it's going to degrade to sulfate. That's going to become acidic. Just look for a pH change. That'll fix it. Okay, that's a good idea. And then we had some other people said, oh, no, don't look for that. Look for COD, so oxygen demand. That'll tell you right away, directly proportional. So we had all these kind of good ideas from the field, from our operators at the plants who thought this was going to be pretty straightforward, and we just really needed to validate that their particular perception was correct. Now I'm going to let Paul kind of jump in here because he did the lion's share of this work and talk about what we actually found.
Sure. Deanna, can you hear me okay?
You bet.
Perfect. Yeah, so just to build upon the fact that we had a couple different synthetic waters, we'll dive into the surface water first, but this is kind of the typically the analysis that we did for all the synthetic waters that we created. So let's take a look at the pH. Similar to what Deanna had mentioned, we were under this impression that pH might actually change with increasing sulfolane concentrations. So we spiked in sulfolane at various different concentrations, and we did not see any change at all with the pH. In fact, hardly anything. That was just one parameter. So right off the bat, we thought, well, there might be other parameters that we can actually take a look at, including chloride, TDS, and sulfate. This was interesting. I mean, we had a huge range of sulfolane to look at. What we found is that, of course, we didn't see any change between 0 to 10 ppm of sulfolane. Suddenly, there was a shift. There was a change in sulfate and total dissolved solids. That is totally unexpected. We were questioning it, but we didn't see any change in chlorides. So there's been this interesting relationship that happens in the shift right around that 10 ppm mark of sulfolane. It's something to explore further, I believe. So they raise a lot of questions. Not only there's some shifts between the behavior of the sulfolane with different parameters, but also at different concentrations. We go back to the mechanism of which we always believe that sulfolane is degraded to. Sulfolane is typically degraded using oxygen that undergoes kind of a mineralization process, producing carbon dioxide and sulfate. And so we would expect to see there's some sort of relationship between sulfate and sulfolane. And unfortunately, we just don't see that, especially within this experimental design. Do you want to go to the next slide? Oh, perfect. Yeah, questions. The mechanism, excellent. There it is.
Sorry
going
about that.
from sulfolane to, oh, no worries. But yeah, in essence, we are looking for sulfate because ultimately that's what we were always told that it would happen. We'll go on a couple presentations later in this session to explore that as well.
And I will add that we ran these at different time steps, so we could actually we did see the sulfolane reduce in the initial concentration. So we spiked it with 10, but we let the sulfolane degrade until it was almost non-detect. So it's not like we just put it in and there was no sulfate right away. We actually watched the sulfolane concentration go down, but we did not see the corresponding increase in sulfate that we were expecting.
Yes, good point. Yeah, 100%. Right off the bat, there's a lot of different things to look at for chemical analyses. Typically, we've been hearing about this chemical oxygen demand and relationships with sulfolane. So we wanted to explore that further. In addition to that, we recognize that sulfolane contains carbon. It has some inorganic properties. So we thought, well, why not do both the dissolved inorganic carbon and dissolved organic carbon? what we found was actually quite interesting. There was hardly any increase or decrease with dissolved inorganic carbon as we increased sulfolane concentrations. But we did see some increase in both the chemical oxygen demand and dissolved organic carbon. We did see at certain points that exponential increase after 10 ppm of sulfolane. So it's not a linear relationship that we were always kind of told to believe, and this is more of a non-linear relationship, so to speak, after concentration of a much higher concentration of about 10 or so. So from 10 onwards to 150, we do see that increase. We did actually compare, so we'll switch to the next slide, Deanna, about the pH. We did actually compare the different water matrices, of course, between surface water and clay till, which is what we classify as a hydrostratigraphic unit A, and also the fractured bedrock as hydrostratigraphic units C, D, and E. So when we compare the water matrices and see whether or not pH changes, you know, with increasing sulfolane, maybe it changes in different water matrices. We didn't see that actually. We didn't see any pH change in no matter what water matrix we used. So it just kind of confirms that there's no indication that pH would be a good proxy parameter, and especially in the field. Conversely, we looked at chlorides, in fact, the next. So we did see a similar trend, you know, really hardly anything happening in surface water. Conversely, we didn't really see a lot in the fractured bedrock, HSU, C, D, and E. It roughly maintained around 250 ppm. We did see a slight decrease, however, in chloride, but it was not really that significant, but rather at a concentration much higher of sulfolane, we did see that decrease. So it's really only those high-end concentrations that we ever see a difference, and that could be because they're interactions.
Yeah, and if you recall back, that's about, you know, the 10 to 25 is when we started to see changes in TDS and sulfate on that first slide in the surface water. So it's kind of similar type issues with chloride. It's not till you get to really high concentrations that there must be some other interactions happening. And I will try to move that along.
Perfect. So, you know, we didn't really see a whole lot of dissolved inorganic carbon. We didn't really see a lot of interactions or behavior or any increasing or decreasing trends. And so we decided to kind of focus our attention on COD and DOC. Well, what I found interesting is that inherently the matrix itself actually contributed to the COD, you know, bicarbonates and other inorganic materials that can contribute to the COD load. And so we do see right off the bat differences in elevations of COD depending on the water matrix, of course, that we use. When we added incremental sulfolane concentrations to each water matrix, we didn't see a shift until only, again, later on when we reached higher concentrations of sulfolane. We actually saw a decrease in COD when we looked at the glacial till or the clay till. That was something that was unexpected. We always thought they would be kind of increasing similar to what we saw in fractured bedrock surface water, but we saw the opposite. Dissolved organic carbon is actually quite interesting. We didn't see a lot of contribution, obviously, from the water matrix itself. This is, in fact, looking at organic carbon, not inorganic. So we wouldn't expect to see any organic carbon other than the loading of sulfolane itself. So we didn't really see the contribution that we would have seen in COD. We did see an exponential increase no matter what matrix we actually used, and that started roughly around 2.5 ppm of sulfolane. So that might be actually a parameter of interest going forward, but yeah, we obviously see a lot of different variations with the relationship of different parameters chosen. So yeah, Deanna, do you want to go to the next slide? You want to take it from here?
You bet. So what were we hoping? We were hoping that our field guys were right, and we were going to find something that was pretty simple that we could use it with an indicator strip or something we already had, something cheap that we could take in the field and measure and say, okay, if we've got, you know, if we're in a surface water and we have COD over a certain amount, there's probably over background, there's probably sulfolane there. The answer was we didn't find anything like that. We didn't find clear correlations. Moreover, the range in the replicates was quite large. We didn't get, we seem to have certain cutoff criteria. It was 10 milligrams per liter, pretty obviously. In the surface water, it was about 50 milligrams a liter in some of the groundwaters. We did see that sometimes in one type of water, sulfolane caused a parameter to increase, and in a different type of water matrix, it decreased. So yes, it was validating how complex these reactions were, but it didn't help in finding a proxy parameter that we were going to be able to use as a cheap and easy way to say, yeah, we have a lot of sulfolane in this water, or no, we don't. Keep drilling, step out. So, you know, it was not the most successful of projects in terms of achieving its objective, but it did help clarify that there's more work to be done here. And really what we want to focus on next is trying to find an indicator that will work. So we've ruled out the easy stuff, and now kind of the next steps are, okay, well, if that won't work, what might? And I think that was it for this first presentation. So we are taking a break. If anyone has any questions, happy to answer.
Thank you, Deanna. Thanks, Paul. It's very interesting, very cool study design, and the synthetic water is certainly novel to me, and good for you guys for trying to track that down and create these different water matrices. I do have a couple of questions in the GoTo platform, and anybody else who's thinking of asking a question, please do type it in. I'll start with the two that we have. One person asks, do you have any thoughts as to why COD might be different in the till?
Yeah, the reaction of COD in the till unit.
Yeah, I believe that's,
Yeah.
I think that's the question.
So I actually, so part of what we thought was that the sulfolane actually ended up having a chemical reaction with the clay because of the charge that was causing some sort of interference. That was the original thought. I don't have a good answer. We saw it over and over again, though, that as soon as you got 2025, there was some sort of physical reaction between whatever was going on in that clay water, which I'm trying to get back to what you can see for what's in it, because it's pretty high in sulfate, as I recall. Paul, I don't know if you have a better answer. I know we talked about it a lot, but we never found this clay till.
Yeah.
I mean, there's not a lot there for it to be causing this odd reaction, but we saw it over and over again.
Yeah, you're definitely right. I mean, the exact chemical reactions that might be involved, it's hard to kind of distinguish that, but we did see obviously over and over and over that difference, that decrease. It could be, you know, the TDS or even some of the inorganic ions that might result from that. We similarly tried very hard not to create crystals and scaling when we created the synthetic water, hence why we actually had to create sub solutions. And one aspect is sulfolane does create some crystals. We do know that from previous studies. So that could be a result. Whether that indicates decrease in COD, maybe that's interacting with the bicarbonates. You know, there's a lot up for interpretation there.
Yeah, shorter answer is sorry, we don't know.
Yeah, another presentation.
Yeah.
We have another question that asks, is it a possibility to use groundwater from a clean area of the site or a nearby site and sterilize it?
So we tried that first. That's where we started. and you can't get replicate data. Like sterilizing it didn't help. Every time that it got, the longer it sat open to oxygen, the worse it got. And no matter how much we tried to put it in amber jars or put tinfoil around it, there was so much variability. We didn't get good results. And the only thing we could think of was that it was changes within that water. And when we switched to synthetic, I'm not going to say they're correct, but they're consistent results. And that's not what we had when we went upstream and got some background water and took it to the lab, which is where we started. Yeah.
All right, thank you. We have one more question via the platform. One person asks, did you look at the mineral composition of any of the precipitates that were formed in your work?
No. We actually, the way that we created it, there were no precipitates. Once we got it right, first couple times we were trying to create synthetic water, there was some, but no, there were no precipitates, so nothing to look at. But it's a good idea. We could try loading up real groundwater and see if we get precipitation. It's a great idea, but we didn't have any precipitate to measure.
Got it. We've had one more question come in. Attendee asks, was the form of sulfur in the solution examined, or was it assumed to be SO4?
Great question. It was entirely assumed to be SO4. We did not do a full sulfide analysis of the full cycle. No. Good question.
Great. You're generating a lot of interest. Questions keep coming in, which is great. Thank you to all of our attendees for clearly being engaged and asking really thoughtful questions. I have one more for you, if you don't mind. So why do you believe there was no sulfate generated from the sulfolane decomposition?
So yeah, that's it. I'm just going to get to this. Why do we not? So here's the thing. I'm not saying there was zero. I'm saying it wasn't a good indicator compound. My gut is that there are a lot more daughter compounds and kind of intermediary compounds, and we'll get to this on the next presentation, I think, before you get to full mineralization. So I'm not saying there's none. I'm saying that you cannot use it as an indicator of degradation. And we've tried this a number of times to measure sulfide, sulfate, where I have done the whole sulfur cycle, and sulfolane goes to zero, and I've got no sulfate, and I went 60 days. And so yeah, I don't have a good answer as to why, but it's not measurable. And on that one, we did ICP-MS as well to make sure there was no just sulfur compound hanging out. Can't find it. So that's why I think it's not there.
Very interesting. Well, why don't we pause there on questions for this speech. Of course, as Deanna mentioned at the top, if anybody thinks of further questions relating to this presentation or other presentations as we go, do feel free to keep putting them into our question box, and I'll raise them at the appropriate time. So I'll take my face out of here and let's get back to the good stuff. Thank you very much.
All right, so we're going to move to bacteria, I believe. Hey, Paul?
Yeah.
All right. So there is evidence in other people's research that bacteria communities have been able to degrade sulfolane. And we at Shell we've been using this at a couple of our plants as a biotreatment for quite some time. So we started thinking, how much do we know about this? We know it works. We did a lot of work in the early 2000s. Does it work? Yeah, sulfolane goes away. Fantastic. How much do we know about this, and how do we optimize? How do we know if it's the best it can be or not? And so we kind of threw this challenge to ourselves, and we had some initial thoughts. And the first one is, we know sulfolane is biodegradable. That's been shown by 3 or 4 people, especially Dr. Lay up in Alaska, but for quite some time, There's been evidence that it's biodegradable, and Pseudomonas was always the species that was considered to be the species. Although, to be honest, at Shell we seeded our bacteria population with municipal wastewater seed, and it seemed to work. So why is it working? Well, it must be that sulfolane is a carbon source for bacteria. That was kind of the assumption. So we dug into that a little bit, and we said, and it only really works in aerobic conditions, or works fast in aerobic conditions. Okay. Does it work in anaerobic? The answer was, well, no, because we don't see it naturally attenuating, so it must not be happening. There really hadn't been a lot of studies on that. It was just a logical extrapolation of the fact that in surface water where there's aeration and there's bacteria, no problem, you see sulfolane naturally biodegrade, and you don't see that in groundwater where it's anoxic or anaerobic. So that was kind of the only rationale that we could find documented from that perspective. And then if we want to get optimized degradation, do we really understand temperatures, pressure? Is there a optimal concentration? Is there too much concentration that becomes toxic? How much sludge should you let accumulate? There are all these kind of questions, and really we were trying to look at our existing system and what do we know about it? And so this little light bulb went off. Didn't go off in my head, I wish it did, but it went off. And the question was, well, if sulfolane can degrade just slowly, why are we not trying to find out what bacteria already want to do this, and not trying to come up with seed from municipal and force them to eat sulfolane? So, okay, well, that's a good idea. And then, well, how does it actually work? Well, we don't really know. We just know it does work. And so we went and started looking at the literature, and all of it was bench scale testing. And there was some pilot size testing that was done, but most of it was about how fast can it happen and does it work? Not a lot about how does it work, and temperature, flow rate, any other condition. What do we need? So we started looking. Well, we've got a lot of groundwater that's got a little bit of sulfolane in it. Maybe we should start looking at what's already there. And this is, again, you're going to see this repeat a couple times. Apologies, but this is what most reports say is happening. So this is the how, and it's stoichiometrically correct. This is what we think is happening. We're getting these sulfonic acids that are getting created. And if you look at Pseudomonas, this is an enzyme that's there. So it's a plausible degradation pathway, which also helps explain why there isn't just an immediate degradation of sulfolane and increase in sulfate from the last question, because this is likely going to hang around for quite some time before it degrades. So it's going to be far from my site before it actually becomes sulfate and water, CO2. So it's the last slide I'm going to go, and then we're going to let Paul talk about bacterial DNA sequencing, which is kind of interesting. So there are two things you need to think about when you talk about bacterial testing. One is ATP, so same as in our cells and as in triphosphate, and then DNA sequencing. So what is ATP tells you, is it alive? Is it active? DNA says, what is it? Because it doesn't matter if I imported from the wastewater treatment facility that I took the bacterial seed from, that there's a whole bunch of Pseudomonas if it's all dead. And conversely, it doesn't matter if I've got a whole bunch of active bacterial communities that don't and can't degrade sulfolane. So you need to look for both, and that's kind of an important point. And so we went and said, okay, so we're not just going to we went to one site and we took 11 different wells. We looked at what unit are they in? So we tried to take from the same aquifers for the most part, although we went across a few. We wanted a range of sulfolane concentrations, like up gradient background, never seen sulfolane, to most highly impacted. And we tried to come up with a small set of data that we could run these bacterial assessments on to say, well, forget trying to come up with or add a new bacteria. What if we already have it there and we just need to foster its growth? All right, I'm going to hand it over to Paul here. He can start explaining some of the stuff he was doing.
I'm on mute. That's perfect. Well, thanks, Deanna. I mean, this is really interesting to me. I'm not a microbiologist, but certainly this is definitely an interesting topic. You know, ATP, level of active and living microbes in each sample, perfect. You know, sulfolane has been used and thought as a carbon source for bacteria. So one must think that if there's sulfolane, if there's that energy source present in the monitoring wells, then there should be actually an increase in ATP counts. We didn't see that. We saw quite a variety of levels of ATP right across the board, from really, really high ATPs to really, really low ATP. And, you know, I'm basically kind of comparing well number 1 to well number 7. That is quite a large difference between these wells. There are differences between, you know, when sulfolane is present and when there's not, but for the most part, there was really no clear relationship between when a well had sulfolane in it and the level of ATP. There was not that relationship. So as, you know, an increase of sulfolane, you had an increase in ATP. We did not see that. So unfortunately, similar to the, you know, the previous slide where we were always trying to look for proxies, unfortunately ATP is not one of them. But regardless, we had to figure out what wells actually had live bacteria. And to be honest, we were pleasantly surprised. There was a lot of microbes active, just not necessarily correlated with sulfolane. Okay, so that was ATP. Now we move into even more fun stuff through gene sequencing. And this is kind of a typical result of what we would see. This is a nice pie chart that tells us kind of the percent fraction of the overall bacterial communities. We would like to see different species and overall distribution of these species. Of course, with all these studies and all this focus and all this attention on biodegradation of sulfolane, there was obviously some key species that kind of were highlighted. So automatically we're like, well, does these species actually exist in the monitoring wells? Turns out they actually do. You know, 9 out of the 10 species were actually present in the wells, right across the board. And so this is very encouraging. Not only did we see that there could be this potential of indigenous bacteria to, you know, use sulfolane as a carbon source, that's great. We see this in the groundwater. And going back to the previous researchers in previous studies that have been done before that highlighted the fact that Pseudomonas is kind of this bacterial species that might be responsible. We actually did see that as the most frequently detected and at higher fractions across the monitoring wells. So this was promising. This is something that we want to build upon. But of course, these are not the only bacteria present in the water. We also have to acknowledge the fact that this site is in a sulfur-containing, you know, area. And is there bacteria that might be linked to sulfur? Sulfolane itself has a sulfur component, and might actually be an energy source of itself in that sulfur. We go back to the mechanism of which sulfolane has thought to be, you know, reduced to sulfate. Well, it may not actually end there, unfortunately. And so if you're looking for sulfate, sulfate is actually used as an energy source for sulfur-reducing bacteria. And so even looking at sulfate may not indicate that because it's already been degraded by other bacteria species present. And so our study not only identified previously linked species, but rather also identified bacteria that are both sulfur oxidizing and sulfur reducing. We saw actually these species both oxidizing capabilities and reducing capabilities in the same well, which is also an indication of a very mature bacterial community present. When we try to correlate this with the presence of sulfolane in different monitoring wells at different concentrations, we really could not see that. And this would suggest that, maybe sulfur is not actually used as an energy source. I mean, you revert back to that carbon source being that energy. So there's still some work to be done here, but we want to account for a lot more possibilities of different bacteria contributing to the degradation of sulfolane itself. I mentioned before that there's quite a lot of diversity. And yeah, there was quite a lot of diversity. Many apologies, this is a very busy slide. In fact, we even said that on the slide because, wow, I mean, where did you begin on this? We did see hundreds and hundreds and hundreds of different types of bacteria. And so you can imagine how difficult it might be to select only one of these bacteria species to be actually responsible for sulfolane degradation. We think that, and we highly suspect that there is a collaboration, or rather a community of a mixture of bacteria rather than a single species responsible for sulfolane degradation. Monitoring wells 3 and 4 are the wells that are actually non-impacted. And we do see quite a lot of diversity there. But similarly, we see the same kind of diversity right across the board, just maybe in different fractions of that particular bacteria in the case of Pseudomonas. When we try to take a look at the number of different species present, accounting for the diversity that we find in monitoring wells, and correlate that back to the presence of sulfolane, unfortunately, there was really no trends, no relationships there. Again, we go back to the fact that sulfolane could be used as a carbon source. We think that maybe the diversity and activity could be associated with that high number of sulfolane concentration for monitoring well, for example, 10 at 35 ppm, but we just don't see that. We do see slightly increased diversity in wells that are non-impacted, but not a significant difference. So really, sulfolane may not actually have an impact on the total diversity, but might actually influence the specific type, maybe the overall abundance of that bacteria. Of course, then we had to take a look and account for the fact that these wells are located in different regions of the impacted site and might be actually within hydrostratigraphic units. That might actually lead to some explanation and some relationships there. But we plotted, of course, the number of different species to the HSUs of interest, and those typically are fractured bedrock, gravel till, and deep gravel. We performed a non-metric multidimensional scaling analysis here, and really we're kind of looking at overall the abundance and phylogenetic similarities of all the bacteria in each sample. There might be some trends in there, but there's nothing that really kind of jumps out at us at this moment in time when we compare the diversity with the HSUs. So something that we're interested in and continuing to look at, but at this time, using this study, we didn't see anything that pops out. Of course, we go back to the fact that sulfolane is biodegradable. Well, how do we use this knowledge into remedial activities on site? As mentioned by Deanna before, we are employing this type of bacterial-based approach at one of our sites, and we did see some success with the sulfolane removal. Using indigenous bacteria, maybe we can actually augment an environment to encourage the proliferation and overall increase the removal efficiency of the sulfolane. And so by augmenting the environment to encourage bacterial growth, we wanted to do a study here. The study was conducted over a 6-week period of time, so it's quite long. And we chose groundwater. We spiked it with sulfolane, a known amount of sulfolane at 5 ppm. So hence it's impacted groundwater. We split that up into a treatment stage where we actually added nutrients, ammonium polyphosphate, and also aerated that over the course of the 6-week period. And conversely, in sample 2, we didn't add any nutrients, and we actually didn't aerate. And what we found was quite interesting. We wanted to see how the bacteria changed throughout the treatment process. So first off, we had to do the genetic sequencing of the groundwater. That was quite interesting. We saw a lot of diversity in that water. We spiked it, as you can see in that red dot there, at 5 ppm on groundwater. And over the 6-week period, we took the last sample on the 6-week period. Obviously, sulfolane does degrade much faster than 6 weeks, but nonetheless, we did take the sulfolane concentration there, and we did also run the gene sequencing analysis on the bacteria there. We did see change in the bacteria communities itself. In particular, Xanthobacter and Rhodococcus actually increased substantially in sample 1 and sample 2 in comparison to groundwater. So there was some shifts within the bacterial community as the treatment actually increased and as sulfolane decreased over that period of time. What was fascinating about this study is that we actually proved that sulfolane does degrade in both anaerobic and nutrient-limited conditions, as well as aerobic and nutrient conditions. And we do see bacterial shifts in both treatment stages. So of course, there's a lot of bacteria to look at. What we focused on primarily was what bacteria did we actually see increase in overall the percentage of the community. Here I've highlighted a couple of the bacterial cultures, or bacterial species rather, that we will probably be focusing on later in different studies. But Rhodococcus and Xanthobacter really kind of shot out at us. You know, it was pretty pronounced that they increased over a period of time in the aeration process. We do see similar increases in other bacteria, but you know, for the most part, we saw an increase in a large amount of bacteria. So again, I go back to the fact that it's just not one bacteria species that might be responsible for the degradation of sulfolane. Sulfolane did degrade, you know, with indigenous bacteria present in the monitoring wells, and in fact, degraded in both the anaerobic and aerobic conditions. And I'll hand it back to you, Deanna.
Perfect. I do want to specify the anaerobic. It was open to air, so it was not a sealed container because we couldn't replicate an aquifer that way. So there was some, but you know, anoxic at best. We weren't agitating it. We weren't adding any outside air. It was simply the interface between the surface of the container and the open air. So what did we find? We found there was a whole lot more bacterial community than we expected. I think I expected to see a lot of diversity, but not a lot of active diversity. While there's been a focus on Pseudomonas, I think some of the more recent papers are showing that it actually isn't just Pseudomonas. There's a whole bunch of different bacteria that are known, bacterial species, and generally a single genus, but that can degrade sulfolane. And how those different species interact with each other and support each other is the next focus for this. Because while you can create something in the lab, there's a reason for these communities to be present. And maybe we need to start fostering the communities we have instead of trying to create artificial bacterial growing and sulfolane degrading communities. I mean, really, we've been priming our aquifer for quite some time from our gas plant. So we've created a very important little microcosm there that we should embrace. We still don't know the mechanism of biodegradation. We were trying to dig into literature on this and talk to others, and I think a lot of people are just happy it happens, and that's a big nut to crack. So we haven't gone down that way. We have tried to postulate some of the different biodegradation pathways to make sure that we can satisfy ourselves that we at least have some general idea where it's going, which will help us with the bacterial community distribution in the future. And, you know, it may be more beneficial to take the groundwater that you've got from the center of your sulfolane plume and help acclimatize that bacteria and kind of give it a little care, some nutrients, a little warmer temperature is always nice. And focus on helping that instead of bringing in imported bacterial communities that may or may not be ideally suited to degrade your sulfolane. I think that was all we had for this presentation, unless there are some questions.
Thank you, Paul. Thank you, Deanna. Very interesting, and certainly you've had a lot of reaction from our attendees, which is great. So I'm just going to jump right into the questions. Some of them came in a little bit earlier in the presentation, so you might tell me that you've covered it, or you might have another point or two to add. But let's jump right in. One person asks, how does the temperature and availability of oxygen affect the rate of sulfolane degradation by bacteria?
We haven't done a availability of oxygen other than to say aerated versus non-aerated. So I don't have a great like kinetics curve for that at all. But as you can imagine, temperature is always going to have an impact. So the warmer it is, the better and faster you will degrade. So we did an outdoor study, and our system is indoor in a very warm building. It's quite uncomfortably warm unless it's winter, and that really does help with our bacterial progress. But we compared RW9, yeah, we compared water from roughly the same concentration inside and out with the same bacterial, the same everything except temperature was the same. Airflow was close enough in aeration. It was what, Paul, 15-day difference?
Yeah.
15-day difference. So temperature increase helped us a lot. We haven't done a lot of study on that, but we certainly did compare the two, and it was significant. It was half the residence time.
Interesting. Another question asks, is the assumption that degradation is occurring in water over microbial communities acting from mineral surfaces?
Good question.
That is a great question. I don't know. I mean, I don't know. We've argued this a lot of different ways. I think with vegetation, we're kind of augering in, but you don't need to have any mineral surface to make it work. We've tried the MBBR, so the little plastic containers to adhere your bacteria to something. Didn't see any significant increase or decrease in residence time or increase in effectiveness. So I don't know, but I don't see a lot of difference between water itself and then adhered to a surface of some kind. But I don't know.
Maybe I'll add to that. And
Sure.
there's a lot of macro and micronutrients on the inorganic level that could be used as well. So maybe in terms of the geochemistry of the water itself, that might have an impact for sure on the proliferation of the bacteria. But again, I'm definitely with Deanna on this one. We didn't really see, that effect of bacteria sticking onto a mineral surface having any impact on sulfate degradation. So.
Got it. Great. Thank you both. Paul, there's a microbiologist in the audience who asks, as you mentioned, ATP tells you that bacteria are alive. DNA sequencing tells you what is there, but have you considered looking at which genes are functionally active in the presence of sulfolane? And the microbiologist offers to have a chat offline with you about it, but certainly if you have any thoughts, that'd be great.
We've talked about that, and
yes.
I encourage you to look out. We have talked about that a lot. So if you want to reach out, we are welcome to hear it, because we've heard mixed conversations about that, and I'm not the expert, so I'd love to hear from you, and so would Paul.
Yeah, definitely.
Great. That's a nice spin-off from this, and hopefully you guys can have a nice conversation offline. Another person asks, how variable was the general water chemistry between the wells? And if there is, could this be a significant factor in different bacterial speciation and distribution across the site?
Good question. So that's why we had taken multiple wells from the same type of water and sulfolane impact. So yes, there was variability, but we had multiple wells from very similar water, i.e. chemically indeterminate between them, that behaved differently, significantly differently.
Great, thank you. I'm going quickly just because there's great interaction through our question portal, and there's still a few questions that I'm hoping we can cover in the next 3 or 4 minutes before we move on to the third topic. So another person asks, could adding a source of oxygen to the aquifer also be beneficial?
Yes, I would think it should be. I mean, it may have other impacts if you have high iron or something that create unintended consequences, but in absence of that, oxygen always seems to help with sulfolane degradation. Hey, Paul?
I would agree with that for sure.
Great, thank you. Are the intermediary degradation products in the conceptual breakdown path detected in the samples that you were working with?
That's next.
Good question.
Yeah. That's another thing. There isn't a commercial lab that'll look for the compounds that we postulate maybe there, and other researchers have postulated. So we're working on that, but it's a non-commercial lab adventure. So we haven't. No, we haven't. We intend to, but we haven't.
It's also like finding a needle on a haystack. I mean, where do you begin,
Oh.
right? There's probably so many different combinations, and they're also probably intermediate, right? They're intermediate for a reason because they're unstable. And so trying to find that, and even if you do find it, how do you relate that back? Like, it's extremely challenging. I don't know, but definitely on it. We've thought about that a lot. But,
yeah, that's Pandora's box a bit, but yeah,
yeah.
if anyone out there has some great ideas or has looked for some of these compounds, please let us know. We will help, and we'd love to hear it.
Yeah, great quality of questions. Another person asks, did you get any idea about the rate of degradation from your study?
We didn't do actual kinetics, no. So we did one study with kinetics, but this particular study did not have any kinetic component to it. So the rate, no, we looked at overall residence times, but we haven't done the rates yet because we were trying to optimize first and then look at kinetics.
Mm-hmm.
Got it. I'm going to get through the last two here, and then I'll try to leave some time for the next couple of speeches. But we'll get back to this topic if there's interest and others have more questions. One person asks, is there an opportunity to better define who is doing what with tools like positron emitting isotopes and then bioaugmentation in a reactor, since many sulfolane contamination sites are being treated by pump and treat?
Yes, there's an opportunity. We should be doing more collaborative work in that space, and I would encourage. We were doing that when things were less dire in the industry and pre-COVID, but yes, there's certainly an opportunity there that should be revisited. Agreed.
Great, and I'll take this one as the last one, but another person is happy to speak, and they say they completely support your microbial community theory, and they ask, have you evaluated biofilm production and the introduction of macro micronutrients to anaerobic condition tests? And they'd be quite happy to take this discussion offline with you as well.
Take it offline. We have certainly, that's where we're going is the biofilm. We don't really know how to get there, but we certainly have identified that as a key component in the actual how sulfolane is able to degrade is through microfilms or biofilms. Why did I say? Oh, goodness, too much talking. Yeah.
And the final comment that I've just received is just perhaps adds to your learnings from this study slide that you currently have up. One person suggests that a high-res mass spectrometry scan of the water might help elucidate what's going on before and after degradation.
Like an open GC high-res, possible. Yeah, totally could.
Mm-hmm.
Yeah, if whoever had that idea, feel free to. Our emails are firstname.lastname@shell.com for both of us. So send us an email.
Yep.
Love to talk about it.
Fantastic. Love the engagement. This is fascinating, and thank you all for those who are watching at the office or at home and writing really engaging questions. This is fantastic. I'll stop there, and we'll get to our next topic, and we're going to look at extraction techniques.
Yeah, so we're going to
Thank you.
change presenters as well. Also, Paul's going to take a break, and we're going to bring
I'll be
in
back.
Darina. Yeah. Darina, are you around? Can you hear?
Yes, I'm here.
Fantastic. I'm going to ask you to just introduce yourself, because I kind of did the short and sweet introduction of Deanna and Paul, but maybe you could introduce yourself before we get into the meat of the matter.
So my name is Darina Kuzma, and I got my PhD at University of Saskatchewan in analytical chemistry. So I am really hardcore analytical chemist working in the lab. I am the lab manager for Aqua, which is part of University of Calgary. And we are looking at contaminants in waters and wastewaters mostly.
Awesome, thank you. So I'll do the introduction, and when we get to the really detailed stuff, we're going to let Darina take over, same as what I've been doing with Paul here. So we've been talking about sulfolane. I talk about it all day, every day. You lucky folks have been listening for a couple hours now, or feels that way. And right now, if you're looking for sulfolane, well, we don't have a field indicator, which we've been talking about. Really, there's a pretty easy process. You collect the sample, send it to the lab, they process it, they do the analysis, they give you results. There you go. What's so hard? Maybe there's some time I'd like to reduce in this piece here, but otherwise, we've got a pretty good system in place. The commercial labs do a great job of giving us reliable, good samples that we can use to update our conceptual understanding of the sites and meet the regulatory requirements that are in place in our province. So what do you do if the limit of detection says less than detect, but that doesn't make sense to you? Is it really sulfolane is not there? And I'm not talking because it's hung up somewhere. I'm talking about it's been diluted so much, or for example, in one of our cases, we believe that it's discharging to a much larger surface water body and we just can't find it. But we want to prove that it's happening. So how do you do that? Well, the smart way is to concentrate sulfolane prior to analysis. We do that with lots of other compounds. We do that through essentially SPE cartridges, or things called passive samplers. So they may not give you a concentration like when you take your groundwater sample and you put it into the lab, but they certainly will tell you presence, absence. And that's really appealing. So how do you do that? Because there's only so far you can push the limit of detection in a commercial lab or in a lab of any kind. So we got to thinking, well, we use SPE, which is solid phase extraction methods, for lots of other environmental compounds that we want to do the exact same thing with. Would it work for sulfolane? That's the question. They look like these little tubes. They can look like a lot of different things. This is probably the most common, but it's the same concept as air. When you've got a charcoal filter, you're passing air through and you're really trying to see, does anything absorb to the charcoal? And then you go analyze the charcoal. Was it there or not? It's exactly the same. So maybe it's not there in a high enough concentration every day, all day, but maybe you've got a slug of something that works through, or it's really low level, but all the time. How do you find that? Well, we're thinking SPE. So what does SPE do for us? Well, it takes it from being a fairly complex, all those physiochemical stuff we were talking about, it takes all that out, because only the compound of interest ideally is going to sorb to the matrix. You don't worry about all those cross reactions. You can fractionate compounds by class, so you can decide, I only want to look at hydrocarbons, I only want to look at sulfolane, I can only look here. And as mentioned, you can enhance or amplify trace concentrations to prove concept or to prove presence. So here was the question, and there's kind of two parts to it. Can we use it as a passive sampler to say, yes, sulfolane is there in low levels, and we put it, say, in a creek and we leave it for 2 weeks and we come back? And then can you, in the lab, use it, conversely, to concentrate up a sample that is low level without having to leave it in the field for a long time. So essentially it changes the method detection limit. So those are the kind of the two ways we looked at it. I'm going to flip over to Dorina here because the next like 8 slides get a little technical, and we'll let her explain it, and then we'll come back maybe for more discussion.
Thank you, Diana. In the world of solid waste extraction, for the environmental samples, we do have 2 main groups of cartridges that could be used. Obviously, there is more on the market, but for the environmental as water, these are the main ones. And they include reverse phase, where we load the water matrix, and usually we elute an organic solvent. These work very well for moderately polar to non-polar compounds, and the most common ones are C18, so obviously long chains of hydrocarbon. Hydrophilic, lipophilic balanced copolymer, and ENV, which is a polystyrene divinylbenzene type of polymer as well. On another note, we also do have an ion exchange, and these work a little bit differently. They rely on the interaction of charged particles or functional groups on the sorbent itself, and then also charge groups on the analytes of interest. So it's a cation and ion exchange, and depending on the strength of the interaction and the pH range we're working at, they're either weak or strong or somewhere in between mixed ion exchange cartridges. We can flip the slide. Yeah, thank you. When we look a little bit closer at HLB, so this comes from Waters, and HLBC is one of the most common cartridges to be actually used in environmental analysis, just because it does work for a vast number of analytes. It does have a backbone, which is pretty non-polar, but it does have a hydrophilic part to it. That's why moderately polar compounds are retained quite well on this. And then around the HLB, which is in the middle, we also have the analogs of the HLB cartridges. And when you look at them, they kind of contain the same structure as a base of it, but they do have charge particles or charge functional groups on them. So they're a little bit more selective to compounds that could be charged prior of loading the cartridges. The range of the pH we can use this is very broad, as you can see. When we look at the sulfolane molecule, which just contains the non-polar part as well as the polar part, it is not a very clear which of these cartridges we should choose. It's not very intuitive before we try it actually out. The pKa dissociation constant is very large, but that number is not mentioned anywhere in these schematics. We either have a lower number or a higher number, but nothing close to 15. So I think we should just give it a try. The key objective of this study was to test whether we can actually use the solid phase extraction, and if so, which sorbents would be good in retention of sulfolane from the samples that do contain low concentration. We also want to look at the sample matrix. It's quite easy to absorb from molecular water, but sometimes where there is more than just one compound, that could pose challenges. And as I already mentioned, HLB is really good in extracting or retaining a lot of compounds, which is a good thing because it is used a lot, but it's a bad thing because it is not very selective. And then if we are successful in retaining the sulfolane on any of the cartridges, how can we get it out of the cartridge? We need to get it back into liquid phase in order to analyze for it at the end. So the overall process of the solid phase extraction is obviously you get the environmental sample. We do spike a surrogate or something that of a known concentration, usually our compound of interest or something that is very close to it. We load the sample on a chosen stationary phase. So this depends on the analyte and the matrix we're working with. And ideally, our analyte retains on the cartridge at 100%, and everything else just passes through, does not get retained. After we achieve the loading stage, we elute the sample, usually with an organic solvent. we get it back into the solution or a liquid phase to be analyzed. And in order to achieve pre-concentration, so we do increase the concentration by eluting with smaller volume than what we started with, but also we can achieve a little bit of a cleanup in order to remove interferences. For first phase of this experiment, we were trying to find out if sulfolane is actually being retained on any of the cartridges. We did test 10 different cartridges. So we took environmental sample, with the surface water, and spiked it with sulfolane and loaded onto these cartridges. In the first step, we just collected what passed through the cartridge. So everything that was not retained on the cartridge, and we were looking for measuring sulfolane in this collected filtrate. The amount of sulfolane that is retained on the cartridge, ideally we want to have 100% or something close to it, is really just the difference between whatever was loaded onto the cartridge and whatever did not get retained, so just pass through. When we look at the results that we got, some of the cartridges like ENV and C18 did actually retain a considerable amount of sulfolane. That's not actually bad retention, so 60 to 70%. The strong ion exchange for anion and cation did a little bit worse job, 30 to 40%. So that was not as successful. But then when we look at the rest of them, the HLB as a backbone one, and then the analogs of HLB with the charge functional groups did retain close to 100 or 100%. The same was true with the charcoal, and I wasn't very surprised. Charcoal kind of does absorb everything in this world. So the HLB structure seems to be the most promising in order to pursue this extraction further. So looking closer at the HLB and the analog of the HLB cartridge, as I already mentioned, we were looking, we say it's really easy to absorb from milky water, which is very clean, and you really only have your analyte of interest. But what if you have co-contaminants, if you have something else that could interfere with the analysis? So that's why we did a really double experiment with the milky spiked with sulfolane, but also the surface water spiked with sulfolane. And honestly, for these cartridges, we didn't see much of a difference. It was minimal, was not significant difference for the experiment we did, which is really promising because that means either the contamination of the surface water is really low, or it's low enough to interfere with the retention of sulfolane on the cartridges. So for the next step, when we are successful loading this onto a cartridge, we need to get it out. And charcoal is one of the problematic ones. You can load it, but you can't get it out most of the time. We chose the most promising cartridges in this case, and it was HLB, medium cation exchange and medium anion exchange. Why we picked those two over the weak ion exchanges is because there was some additional experiments done on this, and mixed cation exchange and ion exchange showed a little bit better results in retention, just a little bit a higher percentage, although it was a very small difference. Still, it was a little bit noticeable. And HLB was again one of the things about HLB is that it does absorb a lot of things, and so this one should be promising as well. The one step we did in this experiment was to see if we can actually elute this with water as a solvent. So we loaded a very small volume of the spiked, sulfolane spiked water. And then we applied a little bit of milky water that had no sulfolane in it, and measure how much of sulfolane was actually released from the cartridges. Interestingly, that was observed that HLB pretty much didn't remove any of the sulfolane just with water running through it. So that means that interaction between the HLB the main molecule of the HLB and the sulfolane tends to be pretty strong. However, in case of the mixed ion exchange, significant amount was recovered from the anion and cation exchange just by running water through this. So that really suggests that there is an interaction of sulfolane with the functional groups that are put on the main molecule of HLB, but that interaction might be just not as strong. And sulfolane is very soluble in water. It is very attracted to water. So maybe just water is the better medium than the interaction between the cation and anion exchange and sulfolane. And I'm going to pass it to Diana.
Thanks, Tarina. So, sounds great, except here's the first problem. If we were going to use this as a passive sampler, and we can load the sulfolane in with water, but we can also elute the sulfolane off with water, how do I know when to pull it out? So that was not the best result. However, HLB at least didn't have it, so we still had something. But finding that balance between retention and elution is going to be critical if we to develop a passive sampler, because what we don't want is to have elution and retention happening at the same time, and we don't know it. And so, yes, we're loading in sulfolane, but we're also eluting it right off. So, as Darina said, that kind of knocked out 2 of the 3 top competitors that we thought we had. So we started out with 10. We had 6 that worked really well, and then we were down to 3 that we were going to do more work on, and now we're down to 1. So we'll get there. At least there's 1. So the next question was, okay, well, if we don't put more water through here, we just use the HLB, and we use a more typical elution parameter like toluene, what happens? We already know that if we just add plain water here, the eluent doesn't have any sulfolane, so that's good. What we did find was we could not only recover a very large component of the sulfolane that had been loaded onto the HLB cartridge, but we could concentrate it. And in the experiment that we did, it was up to 5 times. I think, Darina, that we probably could have done more concentration had we wanted to spend more time on that. I'm not sure what the limit of that is, but I think it's probably higher than 5.
Yeah, we can absolutely look that up. Yeah.
Yeah. So this is a good thing from my perspective for a couple of reasons. So how do we apply this to passive samplers in the field? Well, we're going to be doing a demo trial next year, as soon as the snow and ice is gone, on can we make this work with HLB? Can we load it? Can we prove our concept? That's important. It also means if you get, and I don't know if anyone's had this problem, but certainly we have had this problem, where you get a sample that has matrix interference and you're fairly low level and you're not sure if the number's real or not real, and you've got a stakeholder who's concerned and you don't believe the number's real. This would offer a cleanup of the sample that wouldn't be degrading the sulfolane, that you could potentially utilize to get a better idea of those low level numbers. Are they real? Are they not? And no, you don't have to do that all the time, but in problematic times when you really need an answer, it'd be nice to have something else. And then the other thing that this could be applied to, at least in our opinion, is potential remedial technologies. If you can load sulfolane onto whatever particular media you're talking about, it doesn't have to be in a little tube for an environmental sample. Are there ways that we could use that as an in situ or ex situ treatment methodology to remove sulfolane from water without having to have residence time, bacteria, all the other components that we always have? Still work to be done on that, but it certainly is an option, especially in areas where, maybe you have a very small surface area that you need to get significant cleanup from. I don't know, but I think what we proved here is is that there is potential for the use of typical SPE cartridges and SPE materials to be used for sulfolane in both removal and potentially concentrating up samples. So I believe we went through that one faster than I expected, but that's all we had.
Thank you both. Very interesting, and thank you for walking us through both the practice and the analytical chemistry that underlies that. To those who have questions, please feel free to type them into our chat box through our webinar platform. I do have one question that's been asked so far. How do you relate the concentrations that you're finding back to the original monitored water concentration?
That's not an easy question. So what you can do for surface water, you can take a flow meter and put, so you put the cartridge inside a secondary containment, like usually a PVC tube of some kind, and have a velocimeter that runs through it, so you know the flow, and then that way you can go back out a concentration. But that is a challenge for all passive samplers. They really just give you a mass, not always a concentration. Can be done, but not as reliably as I'd like.
Thank you. I'll ask a question. I mean, I'd love to hear a little bit more about next year's field demo. I don't know how far into the concept you are, but is there any further information as to what methodology you're thinking of setting up, and what you, I mean, we know what you hope to find, but any other information that that would be interesting to this group?
We're in early days. I mean, we're working with our consultants who have been working on getting the understanding of the surface water bodies so that we can try to set this up for success. Some things we haven't worked out is what's the retention time that we leave it in situ. We still have some work to do to auger in on that. But the idea is to place essentially 5 or 6 passive samplers in locations where we have lots of historical surface water quality data, and then try to back out whether or not we see sulfolane where we would predict it. But early days on that. We got a lot of work to do between now and then, but yeah. If anyone has suggestions, again, just send us a note. Always open to those.
Great. Well, that's it from the questions perspective. So thank you very much for that talk. Very interesting, and we'll transition to our last sulfolane talk. I believe we will get Paul back involved, and we'll hear about co-contaminants.
You bet. Thank you, Dorina, for your support on that one. Very much appreciated. Okay, Paul, are you back?
I am here. Can you hear me okay?
Loud and clear.
Perfect.
Okay.
Last but not least,
Last but not least, some of this is a bit repetitive on the first slide, so I'm not going to spend too much time. Hopefully people who are here are staying, but who knows if you've seen the other one. But sulfolane's a fantastic solvent. Low volatility, high stability, likes aromatics, soluble in water, soluble in hydrocarbons, more dense than water. Fantastic solvent. All those things that make it a fantastic solvent make it really hard from an environmental perspective, but fantastic solvent. And it's not just used in oil and gas, okay? Yes, we use it to sweeten our gas 100%. It was actually created to separate aromatics from aliphatics from condensate. So it was created in the 1950s to pull the more expensive and valued components, generally the BTEX compounds, out of condensate streams, and did that very well. It's only kind of late 70s we started using it for sweetening of gas, but it's used as a polymer plasticizer. It's used in the tanning industry. It's used in paint and as a solvent in paint manufacturing plants. It's used in electrical. My favorite example is lithium batteries. Everyone's all over the lithium battery. It is the complex that's made with the sulfolane compound that allows the electron coupling to happen. And if that didn't happen, then you wouldn't be able to use it as batteries. So, you know, we talk a lot about sulfolane related to oil and gas, but there's very little conversation about any of these other industries that use sulfolane quite a bit. Sulfolane, yeah. So it tends to like to be operated between 60 degrees and 120 degrees C. It can work much cooler than that. It's just less effective. That's a good thing though, because most of our natural gas, and when you pull sulfur out of natural gas, you want it hot anyway. So the fact that it can operate at 60 to 120 as its preferential range is great. It is stable up to 245 degrees, so it's hard to mess it up. Essentially, BTEX compounds, we've just got nice little pictures of what they all are there. That's what that sulfolane was intended and designed to remove. This is how much it actually pulls out. So if you have a condensate, and it will pull out almost 60% of the benzene that's in there. It loves benzene. It's very effective at pulling it out, which made the chemical companies back in the 1950s really happy, because benzene was in demand. Toluene, very similar in structure to benzene. It pulls out quite a bit of the toluene, not all of it, but you know, a significant amount. Step change for ethylbenzene. As you add more carbon chains, the less affinity there is. And we couldn't actually show anything meaningful with these little dots down here, so we just made a subgraph. So essentially, xylene doesn't really extract much. They're not as happy to be pulled out by sulfolane. For whatever reason, the less appendages, the better from a sulfolane perspective. Well, how does that look? I think we've talked about this before in previous presentations, the stacking of compounds, which originally was found because of the volumetric change that happens when sulfolane and hydrocarbons meet. When sulfolane and hydrocarbons meet, the volume required expands by 80-fold. So it's kind of like it's almost like a phase change in the volume. This caused a lot of problems in the industrial design because it wasn't anticipated. And so the way they found out that this stuff was happening is they would mix sulfolane and hydrocarbons, and all of a sudden something that needed a volume of 1 liter now needed 80 liters, and nothing else had changed. So they added 1 liter to 1 liter and got 82 liters and went, uh-oh. That's how we figured this out. These are what the molecules actually look like, nice and stacked, right? So, and I like looking at this. I think it makes perfect sense. I hope everyone else thinks it does, but these are really your number of compounds, N equals 2, 3, 4. This is your sulfolane molecule here, right, with your 2 oxygens. Basically what it's showing is that benzene just makes these little happy boxes and stacks around sulfolane, which is why you see that volumetric change. In the proposed diagrams here of how these interactions happen, you will also see that the more extra carbons you put on, this is orthoxylene, the harder it is for that stacking to be as neat and organized and frankly as strong. So it does help explain why benzene has such a high affinity and orthoxylene not so much. This is not nearly as strong a compound as this nice stacked cluster over here. We believe, and there's been others who have researched it, that these are all van der Waals interactions. And why does that matter? Essentially because it's a really weak bond. And so part of the complexity with sulfolane is that it has these, it can be extremely strong bonded to something and extremely weakly bonded to something, depending upon little micro changes in the rest of the chemistry around it. And so it makes it really difficult to predict and makes it really difficult to understand the physical behavior of sulfolane in the water if it's not like a nice ionic charge. There's nothing here that you have to break. It's just kind of, it's all opportunistic. It's the best way I've been able to put it. So why do we care? I mean, that's all great, Deanna, thank you very much. But why does this matter if we're looking at sulfolane at our sites? And so the question that we came out to ask was, well, if the entire original intent of sulfolane was to have an interaction between BTEX and sulfolane, aren't we a little worried that maybe that's happening at our sites and changing either how it behaves? I mean, L-napple is less dense than water. Sulfolane is more dense than water. When the two of them are together, where do they go? Do you chase it like an L-napple plume? Do you chase it like a D-napple plume? Do they mirror each other out and it just looks like density of one? I don't know. If they're bound together, when I send that sample into the lab, is the commercial lab going to be able to see sulfolane and BTEX, or is that bond strong enough that no, it's going to see this other compound, which is this cluster thing, and it's not going to look like BTEX and it's not going to look like sulfolane. So what does it look like? So I was a little concerned about that. So we started down this path once again, looking for, can we get someone to help us understand if we can replicate these interactions and then send it off to a commercial lab, what are they going to tell us? There were a lot of problems. Sounds like a very simple question. I think you're starting to learn that not much about sulfolane is simple. Yeah, we had to overlook water matrix interactions. We knew that we were doing it in a controlled lab setting. We knew that hydrocarbons and sulfolane like to dissolve in different amounts, and how do we know how much to mix with each other? And more importantly, how the lab actually analyzes BTEX and sulfolane is different. So it's not like I can hand them one sample, they put it on the same machine, and it's supposed to tell me how much sulfolane and how much BTEX is there. They need different prep, they need different machines. In fact, they're in different buildings in most labs. So there are some things that still need some working out. So I'm going to hand it over to Paul here, his favorite slide, and we will talk about kind of some of the stuff we did and what did we see.
Excellent, thank you, Deanna. Yeah, I mean, as Deanna mentioned, this is a very unique compound that can be interacting with definitely BTEX, and we've seen that. One of the challenges is, using the commercially standardized methods, which is GCMS. It requires an extraction process that isolates sulfolane. And so if we were able to see that, these interactions might exist, maybe using a GC method might actually destroy that connection. And that might not be the right instrument to actually look at this interaction. So we went off on a path. It's a very alternative path, and we use a very alternative technique. It's called FTIR. Some of you might be familiar with that, but in essence, it's looking at the spectrometry of the mixture. And so at different regions, at different wave numbers, we are looking at signals. And so within different ranges, we get different properties and different functional groups. In the range of 2,800 to 3,000, we would expect to see more carbon, hydrogen, alkane groups. And this is where we would see much of the hydrocarbons. Conversely, in the areas between 2,400 to 2,600, we would see some sulfur, oxygen compounds, and even less numbers than that. So we really want just to see whether or not we can actually have an interaction, that we can actually see these van der Waal, weak van der Waal interactions and bonds that might exist. We did use a mixture of hydrocarbons. So, this is a very simple, high level, does it work, does it not work? We didn't really go into specific individual compounds that might result in the mixture. What we found was actually quite interesting, and similar to what we believed is the complexing that occurs in the sulfolane moiety group, which is the sulfur and the double oxygen, almost looks like the rabbit ears. of the sulfolane compound. And so when we look on the bottom left image, we don't see a difference when we see sulfolane added to hydrocarbons. We didn't really see any differences, which suggests that there's really not that much. There's not a complex that would alter that. We would kind of expect to see a reaction or signal that's in between the hydrocarbons and also the sulfolane. But in that range, that's much lower down the wave number between roughly around 2,480. We do see something. And this is actually quite exciting. It was very exciting when we got these results, because it basically shows that there is some sort of a complexing that happens between hydrocarbons. We do see shifts in signal. We do see that there is that combination between hydrocarbons and also sulfolane. We do see that in between signal between the two. So there's something there, right? And we were pretty excited by that. So we're actually able to see that in the controlled setting.
Just going to pop in and help explain what Paul's saying here. So you've got your kind of gray, which is your plain hydrocarbon. You've got your purple, which is your plain sulfolane. Then you got the red, which is hydrocarbon and sulfolane together. And that's what he means by in between. So we would have expected the red here. It just followed exactly like a hydrocarbon. But the absorbance was changed by the complexing such that it behaved more, it essentially behaved differently than sulfolane alone. And that's what he means by in between, just to clarify there, Paul.
Yes, thank you, Deanna. It's hard to see on my screen, I imagine, for others as well. So I appreciate that.
No worries. All right, so really this was proof of concept. Do we actually see complexing? Because if the answer was no, then the experiment was done, right? The answer was yes. So then the question was, well, okay, so we can see it with an FTIR, which is not what all the commercial labs use. So we said, okay, well, let's go to a commercial lab and see what they can do. So the first one, we did 2 studies. The also switched and did just specific BTEX compounds instead of doing just a hydrocarbon blend, which is what we had done with the go, no go kind of component. And I'll go back to Paul so he can explain.
Awesome, thank you, Dan. So there was quite a lot of differences using the analysis just for analyzing BTEX. We did see with and with, there's differences with and without sulfolane, and we expect sulfolane at concentration of 5 ppm. Overall, we saw right across the board that sulfolane did reduce recovery percentages for all BTEX compounds. We did see that difference more so in benzene and toluene, followed by ethyl benzene. Xylenes, again, we didn't really see a whole lot, but we did see that slight reduction. Sulfolane reduced recovery of benzene, like I said before, and xylenes. Now, there's a caveat here, because MMP xylenes, metamparaxylenes were analyzed together. So there's maybe some work to be done there to differentiate that and to expand, but for the most part, we did see that interaction occurring and thus reducing recovery. So when we looked at that, we also had to take a look at what happens when BTEX is added to sulfolane, and now we're looking at sulfolane. So we took a look at that, and it turns out that BTEX also reduced the percentage recovery of sulfolane by quite a bit. Xylene and benzene showed the highest, surprisingly xylenes, reduction of sulfolane percent recovery, followed by toluene and ethyl benzene. So a little different pattern and different trend, but overall we did see that in both the analytical techniques. So that affinity is showing up in both methods. When we compared study 1 and 2, where did we land? BTEX and sulfolane are not analyzed, again, using the same methodology. So it was a little challenging, but we overcame that and did one method versus the other method. When we stacked up the results, we did see a very similar reduction in both methods with benzene. And the rest of the methods showed a little bit different. So xylenes, had a higher reduction percentage of sulfolane in the second method when we're only looking at sulfolane, and that was the same for meta and ortho. Toluene and ethylbenzene, a little bit different as well. But I guess that the main point is that we did see affinities in both methods. It's a little bit different in both, but that's kind of what we expected.
Oh, sorry, go ahead, Paul.
No, go ahead, Deanna.
I was just going to add in, because I don't remember if I recalled to say it, and I apologize if I didn't. We did not use the FID screening for the sulfolane concentrations. We used the MS spec, so that did include an extraction of DCM. So it was not we weren't looking for the high concentrations of sulfolane, we were looking for the lower ones. So before that question comes up, because I should have mentioned that before, and apologies.
That's a good add. Yeah, because I think it was kind of confusing, right? I mean, GC, you could use FID and MS. There's just a lot of analytical techniques there to look at it. But I guess, ultimately, we did see these interactions observed in using routine analysis. We did see the reduction in recovery of sulfolane from BTEX, and that's where we kind of shifted forward. We were trying to actually understand what that might be. So Deanna, if you go to the next slide, we go back to the steric hindrance, and Deanna, you mentioned before the longer carbon chains of these BTEX might actually interfere with that stacking effect. Partially, we're kind of looking into more of a steric hindrance factor that might impede the stacking arrangements. So we're trying to get a better understanding of how these affinities might be occurring within impacted water. So a little bit more work is required for sure. Deanna, did you want to take it away there?
Sure. So we looked at this, and it was kind of like okay, so you proved your concept. Yeah, you can get reductions. Yeah, you can show that BTEX sulfolane interactions exist. And, while all the other journal articles that talked about it were industrial, were they right? Yeah, kind of looked that way. So success, finally. Yeah, we know that BTEX sulfolane interactions in water, we can see it happen. We know it happens. So what does that mean, right? So what? That's kind of the next question. Yes, we saw reductions. Did we see them outside of the acceptable commercial lab numbers? No. So yeah, we see some interference. We don't see anything that's going to change the answer, which was very relieving, I will say. We see interactions. We can prove that the interactions are happening, and we can force the interactions to happen in the lab. So yes, we're mimicking what behavior we believe is happening in our groundwater systems. The good news is that the extraction method at the lab is strong enough to break those weaker van der Waals and actually get the compounds out and extracted properly and analyzed. So it doesn't really affect the acceptable limit that we already live in. So if you were doing, research, yeah, the numbers change. If you're doing commercial lab, the numbers don't significantly change within the acceptable limits that are already reported. So that's a really good news story. It's a very good news story, because I did not make a lot of people happy when I was talking about the fact that we were finding interactions and maybe we were masking compounds. We did have some challenges. The varying methodologies, we only did this in a single water matrix. We didn't do this in multiple water matrix or synthetic groundwater. So I think there's still some work to be done, but overall, it achieved what we wanted to achieve. What's next? Really, the conversation is moving towards, okay, so we know the complexes happen in situ. We can show that. We know that it's not affecting in any significant way the lab results. So that's good. Whew, we're good. At least in water. We haven't proved soil yet. We haven't looked at that. So that's an option. Then the questions that we started with are still not fully answered. So in situ, if you've got sulfolane and a hydrocarbon plume, is the sulfolane in the water? Is the sulfolane in the hydrocarbon? Is it in both? If it's associated and complex to the way that we expect, how does it move in the subsurface, and where do I look for these compounds? Am I looking for a density-driven plume? Am I looking for a floating LNAPL plume? Do I have to look for both? And then at what point would these weak van der Waals potentially break on their own subsurface? So it's happily moving with LNAPL, and then all of a sudden it's not. How do I find that inflection point? So the next steps on this is to really start looking at how sulfolane behaves when it's complexed in the subsurface, which is 2021 and beyond. Do you believe that is all we had for this one?
Thank you very much, Deanna and Paul. Very interesting, and I suppose a good outcome for for industry and for analytics. And we appreciate you walking us through your methodologies and your findings. If anybody has any questions, now is the appropriate time to use the question feature or the chat feature to submit any questions that you might have. Deanna and Paul, my question was going to be about so what's next and what are our gaps, but I think you've spoken to that quite comprehensively in the last 60 seconds or so. So I'm going to not even ask, and it looks like you were so clear that others feel the same way, that there aren't any questions that have come in at this time. So if anybody has further questions, whether it's about this topic or the other three related topics that Deanna, Paul have put together, please do ask them offline. They've graciously offered to speak. One person has asked what your contact information is, Deanna and Paul, and so I notice it's not immediately available on the screen, or if Dorina had questions, but Deanna and Paul have mentioned that first name dot last name at Shell, so Deanna.Cottrell or Paul.Westland at Shell is the appropriate email address, and hopefully you'll be able to connect in that manner.
Yeah, shell.com, not .ca.
.com, perfect. Yeah, thank you. At shell.com, well put. So I'll perhaps put a bow on our panel here a few minutes ahead of schedule, but maybe that'll give everybody an extra minute to grab lunch or catch up on a bit of work over our lunch break. I will extend my sincere thanks to Deanna, Paul, and Dorina for clearly taking a lot of time and preparation to put together 2 hours of really interesting comprehensive programming on their work relating to this topic. And I will express my thanks on behalf of the attendees, and I'm sure others feel the same way. Of course, thank you to ESA for putting on a great program so far, and continuing into this afternoon. Appreciate everybody's time. My name again was Anand Shrivastava. I'm a lawyer at Holmes & Shire Environmental Lawyers, and look forward to connecting with everybody as the panels continue. That's it for now. Thanks all.
Thank you very much.